All right. So again, the question was, what mechanisms would you predict to be in place in order for the cell to ensure that it has a way to make as much ATP as it is using? Okay, what are some ideas? I don't know where exactly the groups are, so you guys got to just, have your representative raise their hand. Joey, what do you got for us?
So, you're going to need some enzymes to control the breakdown of glucose, which is where
Okay.
ATP is produced.
And that's exactly what we're going to spend the rest of our week talking about after today. That you would predict that a cell has a mechanism in place to take fuel molecules. Remember, keeping in mind that a cell is an open system, you constantly have fuel molecules entering into the cell. You expect there to be a mechanism to convert from the chemical energy in the fuel molecules into chemical energy in the form of ATP, right? Because you got to have, if you're going to synthesize ATP and it requires energy, you got to have some way of taking energy in one form and putting it into energy in another. Good, I love it. Even before that, yeah, Daniel. Okay, you need something to, at least to ensure that you have plenty of inorganic phosphate, right? Every time you hydrolyze ATP, you release inorganic phosphate. And so the cytosol is going to have an enormous amount of inorganic phosphate inside of it. And what do you know about the movement of materials? Where do they move? Down their concentration gradient. So if you have a ton of inorganic phosphate in the cytosol, where's it going to go? Outside of the cell. And so you need a mechanism to get more inorganic phosphate. You also would predict that you'd have a mechanism by which you can prevent the movement of inorganic phosphate. Phosphate is a polar molecule, extremely polar. Okay? And what do you know about polar molecules with regards to lipid bilayers? Do they move well through them or not so well? Not so well. So a mechanism in place could be not having a channel that will allow phosphate groups out. You trap them. You lock them up. But then have a mechanism by which you can get more in. Yeah, Sadie.
This whole thing is basically talking about metabolism, right?
Yes.
Okay,
Uh-huh.
So because you're talking a lot about the production of ATP as well as the consumption of it.
Yes.
Are there for whatever reason I just feel like proteins just kept on coming to my brain.
Sure.
As something to monitor that. So
Absolutely.
Things like ribosomes or something that are floating around within the cell that are monitoring. Okay, we're running low on protein that's being consumed, so now we need to produce more.
There are, yeah. Here, let me answer this. Unless there are any other groups that want to make predictions. And then, because I'll answer your question, and it would be a prediction that I would make, and you actually find that to be true.
So
yeah.
You need to have enough glucose to make more ADP than you...
To make more ATP.
ATP,
yeah.
rather.
Mm-hmm. Mm-hmm.
Because you use ATP to gather what you need to build the ATP.
Yep. Mm-hmm.
So you'd have to have more energy being made than what is being used.
Sure, yeah. Knowing that processes are not perfectly efficient, you need to have a way of ensuring that you're getting a lot of fuel molecules into the cell, right?
Right.
And our primary fuel molecule is glucose. And so you would predict to have a mechanism by which you can get glucose into the cell. And you know what's really neat is as soon as glucose enters into a cell, it's converted into something that isn't glucose. So that glucose continues to be really deficient inside the cell. And what do you know about the movement of materials? They go from high concentration to low. And so if you always have very little to no glucose inside of the cell, glucose is always going to move down its concentration gradient. But glucose, and we saw this on our exam. I mean, I saw it on your exam. You may not have noticed it, but there was a question on there that said which of these would be most likely to move without requiring energy. And one of those choices was glucose, or without requiring a protein was the question. And one of those choices was glucose, and very few people chose that because I think you either knew or you knew that one was like, oh, this is nonpolar, so that one's a good one. But you saw glucose and you're like, well, it's got to require some kind of a protein, and it does. It requires a transporter. You have a transporter that binds glucose and draws it into the cell. On top of that, you have insulin that acts as a co-transporter that as insulin moves down its concentration gradient, brings glucose in. Anyways, this is another story for Wednesday. A mechanism, Sadie, to answer your question, a mechanism that I would predict to have in place is a way in which I would predict that ATP would act to regulate the rate of various reactions. Because if ATP itself regulates the rate of reactions, then the cell has a mechanism by which it assesses how much ATP is there. And what you actually find, and we'll talk about this on Wednesday and Friday, is ATP is the product of cellular respiration. One of the products of cellular respiration. And it actually acts as a negative feedback on the whole process. So if you have a lot of ATP, the process slows down. And so it's a way in which the cell can constantly monitor the amount of ATP present in the cell. Yeah.
So in other words, it's self-monitoring.
Yes. Yep. All right. And then another thing that I would predict, mechanism-wise, wow, this is a lot of stuff. Is I would predict that every endergonic reaction in a cell is coupled with an exergonic. Right? So that every endergonic reaction in a cell is coupled with an exergonic reaction, including the endergonic reaction of making more ATP. That is an endergonic reaction to make ATP. I would predict that that would be coupled physically, not just like in theory, yeah, this has to happen at the same time, but physically coupled to an exergonic reaction. And we'll see that on Wednesday and on Friday. Here's an example of the hydrolysis of ATP being coupled with an endergonic reaction. So this endergonic reaction, this is not spontaneous, right? This reaction here, but this is something that our bodies do to form glutamine, okay, which is an amino acid. Okay, and so this is something our bodies do, and they fuel this by coupling it with ATP hydrolysis. And what you'll find is that the protein that actually catalyzes this, the enzyme that catalyzes this, also hydrolyzes ATP. It actually physically happens on the same enzyme. Those reactions are not just coupled together in time, but also in space. And that's something that I would predict, that you would have endergonic and exergonic reactions coupled in time and in space. Keep that in mind for Wednesday and Friday. All right, next question. What do enzymes do? And this we addressed not on Wednesday, because on Wednesday we talked about research abstract, and then we talked about quiz, and then we ran out of time. I gave you your exams back, and we ran out of time. But on the quiz, it talked about what enzymes do. And so enzymes, basically, they act as biological catalysts. They catalyze reactions. And some enzymes catalyze non-spontaneous reactions. Some of them catalyze non-spontaneous reactions, so the reaction wouldn't happen without an enzyme, period. But many enzymes also catalyze spontaneous reactions, spontaneous processes. They would happen without the enzyme, but the amount of energy required, or the amount of time required for that to happen, is too much to make the cell viable. And so they do this by lowering activation energy, and they lower activation energy by doing this, stabilizing the transition state. So you had a question of how do enzymes actually lower the activation energy on your quiz on Wednesday, right? Do you remember that? Okay. And so they do it by bringing reactants together, physically binding them and bringing them close together in three-dimensional space. They do it by exposing reactions to alternate charges, which increases the instability. And then they also do it by forcing strain, changing the shape of the reactants, making it a lot easier to break bonds. Right? If you start bending something, it's a lot more likely it's going to break. Right? Even if it's unstable. And what do we even mean by an unstable bond? Somebody tell me that. If we say a bond is unstable, what does that even mean? So saying that that third phosphate group bound onto the sugar and the adenine, saying that's unstable, what does that mean? Yeah.
It's not energetically favorable for it to be there in the first place.
Yeah, so that it's forcing electrons into a position that they don't want to be in. Right? So covalent bonds, like that formed between a phosphate group and ADP, are the sharing of electrons. So if that bond is unstable, it means you're forcing electrons into an energetically unfavorable position. And so that when you actually break those down, the electrons can move to a more favorable position, and as they do, release energy. Okay? But what makes it a lot more likely that a bond's going to break? You start playing with it, right? It's unstable, and you play with it, and then that instability actually comes into the bond just breaking. Yeah.
So in general, are like more complex molecules like more energetically unstable?
Yes. In general, yes.
So
I mean, there are exceptions to that rule, but the more complex a molecule is, typically the less stable it is.
okay, so I just think like we talked about how like plants produce like those complex organic molecules,
Uh-huh.
but they're stable enough to get like all the way to our cells before they break down.
Well, I mean, think about, we talk about uranium, like radioactive uranium being unstable, but its half-life is 3 billion years. Like, well, how unstable is that really? Right? It's going to take you 3 billion years to get half of it to release whatever's in the nucleus, making the nucleus unstable, to make a stable nucleus and give you some form of lead. And so you're like, okay, well, it's unstable, but unstable doesn't mean that it's ready to change right at this moment. If that makes sense. Yeah.
Is that how DNA is able to stay? 'Cause
Yeah,
that's
DNA
very
is
complex.
unstable, but it's, I mean, it's, so if you have DNA and you just leave it, it's not going to stay as DNA forever. It's going to deteriorate and it's going to fall apart. And it's because it's unstable. But it's far more stable than RNA. So it's going to last a much longer time than RNA would. RNA is really unstable. RNA is like nitroglycerin, if you know anything about nitro. Anyways, we'll talk about that another time. All right, so here's the idea of activation energy. And so if here is the energy level of your reactants, here's the energy level of your products, you're like, that's an exergonic reaction. That should be spontaneous. But it doesn't mean that you don't have to insert energy in order to break the bond. Right? You break the bond, you're going to release a ton of energy. Way more energy than you had to put in to break it. But you still have to put in some energy to break it. So overall, the process is exergonic, but you still have to input a little bit of energy. Enzymes work to lower that activation energy. And they do that by physically bringing the reactants together, by introducing those reactants to new and novel charge, and by creating some torsional strain, changing the shape of the reactants, putting pressure on bonds. Make sense? Okay. So here's another illustration of just that, but without a boulder. And then here's showing you what an enzyme does in lowering this activation energy. Okay, so here's the initial activation energy. Still an exergonic reaction. You would predict that this would be spontaneous, right? Your reactants have this much internal energy. Your products have this much internal energy. You're like, that's an exergonic, that's a spontaneous reaction. But in order to get this, you need to break bonds.
So it doesn't matter what the process is, it only matters what the result is for the
For energy, yes. Yep. Yeah, absolutely. Yeah, that's a good way to put it. It doesn't matter what the process is. Exergonic just says what is the outcome, right? Is the end going to justify that process? And yeah, it would, because you're going to get out a lot more energy than you're going to put into it. But you still got to put some energy into it. Yeah.
So these reactions that create energy, or not create, release energy rather, seem to be combustion reactions.
Well, there are more than just combustion reactions, but yeah, combustion reactions. So oxidation reduction reactions release energy. What do you call it? Fission reactions release energy. Fusion reactions will release energy. Mm-hmm. So it's not just combustion reactions.
So what keeps these reactions from going crazy and going just really going and going and going and going and not?
The number of enzymes that you make.
Okay.
But you mean, so just because a reaction is spontaneous doesn't mean it's like a chain reaction, right? So if you start making reactant, the rate of the reaction increases exponentially. I think that's what you're thinking about right now. You're thinking about something that's like a catastrophic chain reaction, like maybe a fusion reaction. That once you get started, the rate accelerates throughout. Mm-hmm. That's not the norm for chemical reactions. The norm for chemical reactions is to have a constant rate of reaction. Now you can change the rate of reaction, but it's still a constant rate of reaction. And then you can change the rate, but it's not going to change the rate to now make it exponential. It's just going to change the rate by shifting like where your linear relationship is.
Okay.
All right, so let's talk a little bit about how enzymes actually work. So enzymes do not change delta G. They do not change the thermodynamics of the reaction. Okay, just in the same way you learned in chemistry, if you're in chemistry, that a catalyst does not change the delta G. It just changes the rate of the reaction. And so the way enzymes do this is they actually physically bind the substrate or the reactant. You had a quiz question on Wednesday that said, what are two things that enzymes bind? And the two correct answers were substrate and reactant. The reactants of the reaction and the substrate is something that the enzyme binds. Okay, so they're shaped not just morphologically or not just physically shaped, but also chemically shaped. So that if your reactant has a particular charge on it, the enzyme will have the opposite charge to attract it. And so usually when they bind at the active site, they both change shape. The enzyme changes shape, and as it does, it forces the reactant to change shape. And this is, again, creating instability and creating more instability and getting the reaction to actually take place. Yeah.
That doesn't require ATP, that changing of shape and that binding?
It may, but if it does, it's going to produce more ATP than it would require. But yes, sometimes it does actually require ATP to change its name. Usually not. Usually just the chemistry of the interaction is enough to change the shape.
So, if I could ask,
Yeah.
Enzymes lower the activation energy without actually using energy by putting.
Well, they lower the activation energy, and you're still going to need to supply energy.
Well, I mean, is the way the enzyme works is that it doesn't actually use energy to work as a catalyst, right?
Well, you can't really say that. It doesn't typically use ATP to work as a catalyst, but it usually requires energy, usually in the form of kinetic energy or some other form.
Okay.
Yeah. All right. And so this idea of enzymes is called the induced fit. And so the induced fit is basically this idea that the enzyme forces the reactant into the shape necessary to generate your product. Okay? And that's the idea of induced fit. It's the current model of how enzymes work. Is it possible that it's wrong? Sure. These things are happening on a very tiny scale. You can't snap a picture of it and see what's going on. But this is the idea of how enzymes work. Many enzymes then have elements. If it's a cofactor or that actually influence how they work. So many enzymes have a cofactor or a coenzyme that influences how they work. And it may be that has to bind to make the enzyme function correctly. Yeah, and it's actually, a lot of the times it can work as an on-off switch. A way of turning the enzyme off and then turning it on. Yeah, Emily.
With enzymes, is it once they bind or they whatever they're gonna do, they bind or they attract that other thing, is it done? Do they go, or is it a renewal process like the enzymes?
Yeah, the enzyme isn't used. It's not a reactant. So the enzyme is not used up in the reaction.
So it's still going.
Oh, yeah.
It keeps doing the same thing.
Yeah, but it typically can only bind one reactant at a time. But once it forces that into product, it releases it, and then it binds a new reactant. It's not a reactant. The enzyme is not part of the chemical reaction.
Okay.
It doesn't impact the thermodynamics, and it's not part of the chemical reaction. It's just increasing the rate of reaction.
But our body continues to make more enzymes?
Yes, because enzymes are degraded for other reasons, because they in themselves are unstable, and so they deteriorate over time. And then some of them are deliberately. Like if the cell's not using an enzyme at a particular time, it'll recycle it. And then unless it's like something that's very predictable, cyclical. I like to think about it's the same way I deal with clutter around my house. Like if it's something like a tool where it's like you're going to use it somewhat regularly but not every day, keep that around, right? Just keep it well organized. But if it's something you haven't used in 3 months and it's not like a tool or it's seasonal, I'm not going to use that regularly enough to keep it. I'll get rid of it, and then if I need it, I'll borrow it from somebody or buy it again, All right. And so here's kind of that illustration of what you were asking, Emily. So here's beta-galactosidase, which is an enzyme that hydrolyzes the bonds between glucose and galactose. And so the enzyme facilitates or catalyzes a reaction that's already spontaneous. This is unstable, and so it's going to break down, but this catalyzes the reaction. It lowers the activation energy necessary, creates strain on that bond, breaks it, releases those, and goes to bind another lactose. Okay.
Makes sense.
And it doesn't just bind lactose, that's what this diagram, but beta-galactosidase binds probably any disaccharide. Did you have a question, Grace? No. Okay.
Somebody who's lactose intolerant lacks that enzyme.
Well, I shouldn't say we all. Almost every human being lacks that enzyme, but the bacteria in your gut have it. So if somebody is lactose intolerant, it's usually because their gut bacteria lack beta-galactosidase. Yeah, very few human beings can actually metabolize lactose without your gut bacteria.
Wait, then why is that an easier problem to solve? You just ingest more of that bacteria?
Because it's really difficult to control, to actually get to change your gut bacteria. It's really difficult. It's a pretty involved process. You have to regularly feed on that, whatever that bacteria is, and it's a really prolonged process. It's easier to actually take a gene gun and actually fire it into your intestines and genetically change your gut bacteria to give them the ability to metabolize lactose. It's easier to do that than to change your gut flora.
That's so cool.
Yeah, once those bacteria are cemented in there, they don't want to go anywhere. They don't want to go anywhere. You feed them well. The intestines are a great place to be a bacteria. They don't want to go anywhere. All right, so how do concentration and inhibitors impact enzyme function? This was, again, another quiz question where it just asked you, give 2 factors that influence enzyme activity. So I mentioned it's not a reactant. It's not going to use up. However, it can become saturated, right? If you only have a certain amount of enzyme to go around to bind to your reactant. And so it can actually become saturated. And so we tend to have a linear relationship between the reaction rate and the amount of enzyme we have. So if we have a ton of enzyme, your reaction rate is going to keep increasing because it's not likely that it's going to get saturated. If you have very little enzyme, it's really likely it's going to become saturated, and that's going to be your limiting factor. Most often, the limiting factor is the amount of reactant and not enzyme. Usually you have plenty of enzyme to go around and not enough reactant. And here we go. Because the substrate is a reactant, it is used up. And it can be restored. And this is usually the limiting factor, is the amount of substrate you have. And again, not the amount of enzyme. Usually it's the amount of substrate. This does not form a linear relationship, instead forms a logarithmic. And what we mean by that is if we're going to go on the x-axis, we have substrate concentration. And then over here we have rate of reaction. As you increase your substrate concentration, your rate of reaction is going to increase, but eventually you'll get to a point at which it starts to tailor off. Okay? It starts to tailor off, and that's because you only have a certain amount of enzyme. There's only a certain amount of space for these reactions to take place. Space is limited, the amount of enzymes are limited, and so eventually you're adding more and more substrate, and yeah, it's increasing your rate of reaction, but not noticeably. Not noticeably. And then enzymes can be inhibited by a number of factors. Many of them bind to the same place that the substrate does. We call these competitive inhibition or competitive inhibitors. Or they can be what are called non-competitive, that change the shape. You could also call those allosteric. Yeah.
Aren't most reactions like the reactants themselves, the products themselves are the inhibitors, so that after
Yes.
a while it stops.
Yep, and that's what you find with the whole process of cellular respiration. ATP is a competitive inhibitor of many of the enzymes in that process. And so if you have a lot of ATP, it actually shuts the process down because ATP binds better than the substrate does. Yeah, Joey.
So are the majority of like food allergies based on this idea of like an enzyme, either your body producing it or not producing it, or like that enzyme being ineffective?
Are you talking about food allergies or intolerances? Completely different idea.
They're different?
Yes. An intolerance is you don't have the ability to digest it well, and so usually it creates some kind of a leaky situation. Like you have a lot of this material in your intestines. Oh gosh, what's that fluid component? What do we call that? It'll come to me. But the fluid inside of your intestines, and so you have a high solute concentration. So a lot of water is running into that. If you've got something that you're not metabolizing, you're not breaking down, and it creates like a leaky situation, leaky bowels, and that's what creates some of the pain. And then you can recruit other things. But if you have a true food allergy, you're actually making antibodies against that material. So it's a different situation. An intolerance is often you can't metabolize it, and so it's just building up, and it's creating some kind of a leaky situation. Whereas an allergy, you're actually making antibodies against it. And so you can test for that if you do an allergy test, which are unreliable. But you can test for whether you have an allergy or an intolerance. It's very difficult to test for an intolerance. Chyme. There we go. Well, sort of. So chyme is what's actually in the yeah, I mean, yeah. Yes. I mean, essentially. Yeah, chyme. Thank you.
So for allergies, you can still digest it then?
No, because you actually have antibodies that are attacking it in your bowels. Yeah, that's because most often we have some amount of antibodies leaking into the bowels, and if you've got a food allergy, you usually have lots and lots of issues associated with it. So here's this relationship linear between enzyme concentration and rate of reaction, and here is the substrate concentration and rate of reaction because we get to a point of saturation. Yeah, we're out of time, so yes, can I go back a slide? Absolutely. These cookies, again, are from Rachel in celebration of her birthday and for your culinary pleasure. So go ahead and grab one on your way out, or two on your way out. People have had plenty of time to grab some. Don't grab more than two unless you're the last one to leave, and there's still a whole bunch. Oh, we were so close. So close to getting through all of it.
You ask too many questions.
I know, it's okay.
I can't write today.
Oh my goodness.
Hey Rachel, do you have any plans for your birthday today?
No, I just have classes.
Yeah, I have plans to go to 5 more classes today on my birthday.
Wait, when's your birthday?
I made a huge mistake my entire first year of college. I just packed my Mondays. So like I had both semesters, I had 2 1-credit classes that only met on Monday. And so I'd be from like 8 in the morning until 6 p.m.
Oh wow.
Oh man.
on Monday with no like real breaks. And I did that in the fall and in the spring.
What? Why?
I don't know.
You just thought it would be more productive?
I don't know, but it really, I think it's actually been beneficial for my life because now I feel more fondly about Mondays than I used to.
Wow.
In case you forgot,
Ooh.